A neuron cell is a diploid, right? It has a 2N. Haploid means it's a half of this. Monoploid, they don't use this term, but monoploid may be matching with this 1N. This is also 1N. Tetraploid is a double of this, 4N. Is it two strand? It's two copies, 2N, it's double helix. If you separate them, it's a four strand, isn't it? If it's a single strand, that's a four strand, right? And many of those, most of the problems you guys solved quite nicely. This problem, nobody got the right answer, as I expected. And information amount wise, a single strand and a double strand. Is a single strand is a half of double strand? Well, maybe not, because it's just a complementary sequence in the other side. So basically it's the same information. That's why cDNA library, that's just a single strand DNA. Still that works. Only one side works. The other side is just a temporary, the complementary sequence, which is just a protecting and as used as a template, but that's not new information. When the DNA make a double helix, it forms quite tight double helical structure. And then it is a little compact structure. And then one layer of the distance is 3.4 angstrom. This is kind of a condensed form and complex form. If you make a single strand, they can be three strands, just a random sequence. And it's not condensed, but it's an open structure. If it's open structure, then what's the atom number between one DNA to the other DNA? P to P. How many atoms there? This is a basic atom, right? How many atoms in 1 minute? Or how many bonds in 1 minute? 8. Is it? 1, 2, 3, 4, 5, 6. Another P, repeat up to here. It's a 6, is it? Bond was 1, 2, 3, 4, 5, 6 bond. You guys got the good information of the double helix has 1 unit of 3.4 angstrom, and that's why totally 3 billion, and then you got the number of 10 by 10 to the 10 to the 9. So 3.4 is 3.4 10 to the minus 10. So multiply this is approximately 1 meter. You may have heard it or you got those information as a supporting info. that is working for one double helical strand. But if it is single strand, this is an open form, relaxed form, then what is the length from here to here? Well, if it's a fully random structure, one bond length approximately 1.5 Armstrong. But if it is a zigzag structure, let's assume, then approximately one length here is about 1 Armstrong. Then one unit is 6 bond, about 6 Armstrong. Double of this. Then you can see this is really a compact structure, half size of a compact structure. This is almost double size. So using this as your calculation, then one strand may be about 2 meters. And you have a 4 strand in neuron cells. If you divide them all, then multiplied by 4, totally you have 8 meters of DNA. That's just a calculation for fun, but I wanted to hear a little more creative answer from you. You guys got quite standard answers using double helical strand as the reference. And we didn't count the 2N. Our intention is that not sperm cell, neuron cell, is a somatic cell, which has 2 strands, 1 strand from father, 1 strand from mother. Right? Now, using this as a let's think. If they need the human genome project, which strand they use, you think? From mother side or from father side? Sorry?
Other side.
Because?
Because other X chromosome is one of X chromosome is in
What's the matter? Still, the other X chromosome is randomly expressed. So you just simply sacrifice the one X chromosome. 1 second, X chromosome can be also found on the father side too. Sperm cell can make a Y chromosome and X chromosome, both of them. For human genome project, they used mixtures of the DNA, and it's kind of an average sequence. So many of them has constant sequence, 90.59%, but 1% of them has a different the sequences. And then those sequences will be, maybe, depend on person by person, and also mutation by mutation. Okay. So this was the intention, how I made this problem. You need to think. Last time when I asked this question, some of you, you know the answer about 1 meter. So I want to think a little more. And key point here is, when you calculate those bond lengths, if it's 1 bond, approximately 1 Armstrong, that's the good estimate you can use when you calculate. Okay, bacteria uniquely has a cell wall. Today we started to learn study about the antibiotics affecting some foreign invaders, and we needed to think about what's the difference between the whole body and non-visible. Bacteria also has a cell membrane. Bacteria does not have a nuclear membrane, also doesn't have nucleus, and they have a lysosome, but we don't have the cell wall. Bacteria cell has a cell wall. Plant cell has a cell wall. Yeast cells they have a cell wall too. Okay, membrane structure is a phospholipid structure. Main component of the cell work is there is sometimes this peptide is involved with the too, but if you measure those mass, carbohydrate is the major component. Very interesting. How about the shell of the crab? Crab. What is the main component of the rigid shell of the crab? That's also carbohydrate. It's also carbohydrate polymer. Carbohydrate could be quite rigid, quite a hard material. Very funny way. Okay, how about the penicillin's target is a cell wall which does not exist in our body. That is the target. Okay, that's the target. Cool. Okay, let's see. If you compare cell membrane and nuclear envelope, which one is the more permeable? Cell membrane? Is it? Nuclear envelope and then cell membrane. Nuclear membrane and cell membrane. Which one is the more permeable? Nuclear membrane because? Because of the whole what floor is it? What is the composed of? Nuclear membrane we said is a double layer of bilipid, right? So it's actually it's not closed structure. Cell membrane is a closed structure. It's a fully divided outside of our cell. Nuclear membrane is folded, and then folded form is a contact like this. This is nuclear form. This is kind of open structure. That's why quite big material also can in and out. So that's not closed structure. Okay? Mitochondria is different. Mitochondria has double layer, but outside closed one, inside also closed one. That's a double layer, double closed one. But nuclear membrane is not. Now, I said cell wall is very rigid, but that does not mean that cell wall is really tightly closing. I will show you the structure later. And virus coat. So we don't call virus a membrane. Virus surface structure is peptide or protein. It's kind of a seed. It protects those the genetic material inside. That's why this material is quite well protected and they can survive for long. So they say global warming, if the ice in North Pole, South Pole, if they're melted, then maybe ancient times the virus will come and attack us. That means virus can survive for long because they are well protected. So material transport wise, virus coat, maybe this is tightly hidden inside that coat. That's made of protein, that's why it's called as coat, not the membrane. Okay. So based on this, let's start today's antibiotic study. Okay, antibiotics. Who is this bio?
Bacteria.
Huh? Is that bacteria? Okay. Let's see. So our body is composed of cells. So this our cell is approximately 10 micrometer or 20 micrometer size. And we don't, so the sum of those cells is quite big, but in our body, not only our cells, there's a lot of bacteria living together with us. Bacteria size approximately. Our cell is a 10 micrometer, bacteria size is about 1 micrometer. Some of them are slightly bigger, slightly smaller, but approximately 1 micrometer. Mitochondria is almost a similar size, bacteria size. Chloroplast is also similar size. Now, for a long time, there are some common knowledges, common reason. People believe so. So one of them is, what is the artificial architect on Earth which can be seen from the moon? there's one the artificial architect existing on the surface of Earth that's huge enough, so that's why it can be seen in the moon.
The Great Wall.
The Great Wall is not true, but many people just cite this. So Great Wall cannot be visible from the moon, but it's kind of some expression that Great Wall is big or something like that. So for a long time in our body, same number in our body, we don't know exactly, but depending on the counting, people believe that we have about adult body, we have about 10 trillion cells. 30조. Sorry, 10조. And bacteria number is about 100 trillion. It has been believed over almost a century. And quite recently, other scientists, they recounted and then checked those references. Seems like it's just randomly given number, and they carefully calculated this again. And seems like our cell, if it's 10 billion, 10 trillion, and 100 trillion, that means our cell is only 10% of the total cells. We have 10 times more bacteria in our body. Are we human or our colony of bacteria? Human part is 9 part. But size-wise, human cell is 10 micrometer, and bacteria cell is 1 micrometer. And volume is also, volume is also how many fold per cell? 1,000 times, right? Right? Then maybe the mass-wise, approximately 100 times our cell is bigger than bacteria cells. Still, the bacteria numbers are too many. So other scientists, they tried to count, and then they counted, let's say, red blood cell. It's much more numbers than we thought, and it's a smaller size. So 70% of our cell is a red blood cell, and they count a lot more. So we have about 30 trillion cells. And they counted again this bacteria carefully. Mainly those bacteria, main source is in this gastrointestinal pathways. And then the careful counting seems like the number is about 34 trillion. So it becomes now 1 to 1. So still, it's quite a lot. And not only those the bacteria cells in our body, we have viruses living together too. And some of those viruses seems like it's a symbiosis. They're helping us. Without those viruses, then we have some functional problem. So we have our genome sequencing, genome project. It's sequencing our gene. But as we said, almost a similar number of bacteria live together with us. And which one may be bigger? Genetic information of the genome or microbiome? If it's the same number, our cell is 30 and bacteria is 32, then genomic information wise, which is the bigger information? Our cell may have a 3 billion base pair, bacteria may be smaller, and what would you say? Our genome may be bigger? Suyana, what do you think?
Our body is bigger.
Our body is bigger. Our body, the genomic information is bigger. Do you agree, Jenny?
I was thinking if there's many different kinds of bacteria,
Yes.
then there would be more.
Basically, our cells is basically have the same genome, right? But there may be many different kinds of bacteria in our body. Then if you count them as a different genetic information, this is maybe 100 times or 1,000 times bigger information. They live together inside. And that make, whether we really control our body or the bacteria generate a lot of genetic information, and product of the genetic information is protein or carbohydrate metabolite. And sometimes, depending on those microbiome conditions, our mood is also affected, let's say. So who is really controlling our body, our brain thinking, or our gene, or this microbiome? It's kind of a big study these days. So this is second brain, even they say. Okay, so those bacteria, depending on those shapes, the biggest presentation of this rod shape is a bacillus. And round shape like this, corpus. So if it's connected together like this, like the grape, then staphylococcus. Staphylo means the grape. If it is stranded like this, then staphylococcus. So those are depending on what you shape, we give this is a coccus and this is a bacillus. And these days we hear time to time this kind of term, probiotic. What is a probiotic? 준야, what is a probiotic? 유산균 in English. Do you know 유산균?
Bacteria that comes from digestion.
There's a lactic the bacteria which helps our body. So we take those bacteria which may be helpful to our body. Those are things we call probiotic. Now today we are going to study about antibiotic. Pro means for us, helping us is a probiotic. Antibiotic is opposite. And when I asked who is this biotic in antibiotic, the answer was this biotic, bio is bacteria, is the biotic as an enemy. Then how about here? Who is this biotic? Is it bacteria? This bio is us. For us. That's a probiotic. Right? Similar, looks like it's a similar term using opposite the meaning, but actually the meaning is a little different. So similar way, antibody. Who is that body? What's that antigen? Is that us or that's enemy? That's an enemy. Antibody's body is also an enemy. If you use the vaccine to attack the enemies, that body is also antigen body. Those bodies are bacteria or virus. Those are enemies. Okay, based on this, now we need to think about if we want to selectively attack our enemies, which invaded our body. The best way is we figure out the difference between us and them. Basically, all the life forms on Earth, even including DNA, the virus and bacteria, the basic genetic information is based on same structure, DNA. Exactly the same. Very small minor are the exceptions, but most of those life forms has almost equivalent the structure of genetic information. Those genes, if you take in our body we can make a protein using those genes. So basically we are all cousins, families. Then, without killing our body, and we need to selectively kill the enemy. That's the key. Again, probiotic. So if we take a lot of 유산균, the lactic virus, to help our microbiome and grow them, and you got some infection, if you take antibiotic, and they will wipe out your probiotics. So we don't want to kill our friends or our body, but we want to kill selectively those enemies. Then we need to think about the structure of our body. Our cell is mainly made by this cell membrane, isolate the environment from our body, and based on this kind of phosphodilated. And bacteria has this bacillus with the tail, and they have a cell membrane, and they do not have the nucleus, but they have a genomic DNAs which is spread out in the whole body. And outside of the membrane, they have a cell wall. We don't have a cell wall. And maybe that can be a good target for attacking. Viruses do not have membrane. Sometimes they do. But basic structure of virus, they are inside this capsule or coat. They have genomic information inside here, either DNA or RNA. Outside of those materials are mainly proteins, which can be crystallizable. Very strong, tightly packed structure they can form. Sometimes they call it capsid, like a capsule. So based on this, now we need to think about those mechanism for design of antibiotic. So Kenvin volunteered to present some of those antibiotics mode of action today. So let's invite Kenvin 박수 한번 주세요. 오케이. 마이크.
Okay, welcome back. And hello, my name is Hyunbin Cho, and I'm in Department of Chemistry. Today I'm going to talk about antibiotics mode of action, which is called MOA. And I use propolis every day, and this is also antibiotics too. Antibiotics refer to organic substance produced by microorganisms that can destroy bacteria or inhibit their growth. So antibiotics are substances secreted by bacteria to defend themselves against invasion from other microorganisms for their survival. So they can act as various sites based on the central dogma. The role of antibiotics can be broadly classified into 5 categories. First is inhibition of cell wall synthesis, and second is disruption of cell membrane function. Third is inhibition of protein synthesis. Fourth is inhibition of folate synthesis, and last is inhibition of nucleic acids. And today we are going to point inhibition of cell wall synthesis. First of all, let's see the difference between gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria are stained purple because their peptidoglycan layer is thick, while gram-negative bacteria do not retain the purple dye because their peptidoglycan layer is relatively thin and the dye is washed out. Due to the presence of the outer membrane, you can see antibiotics generally cannot easily penetrate into the bacterial cell of gram-negative bacteria. Therefore, gram-negative bacteria are said to be less susceptible to antibiotics than gram-positive bacteria. The difference in antibiotic efficacy depends on factors such as permeability, the presence of inactivating enzymes, and the target site. And peptidoglycan is compound composed of relatively short peptide linked to a polysaccharide chain, you can see. In particular, it is composed of N-acetylglucosamine, which we call NAG, and N-acetylmuramic acid, NAM, and the main components of the bacterial cell wall skeleton is attached to a peptide side chain. This structure is also known as murin, and the polysaccharide chain of NAG and NAM are arranged in a lattice-like 3D structure by the cross-linking of the peptide side chains. The peptide cross-linking structure varies depending on the type of bacteria. And this is the peptidoglycan synthesis pathway in E. coli. You can see NAM and NAG made it, and they are antibiotics. So we will know 3 types of antibiotics in the synthetic pathway. First is inhibition of cell wall synthesis, and they're commonly inhibition of peptidoglycan biosynthesis is the inhibition, and also it could inhibit cell wall biosynthesis. And the cell wall synthesis inhibition has common factors, which has bacterial activity, and inhibitors do not exhibit their actions on cells that are stopped growing. And antibiotics do not show activity against bacteria that do not have a cell wall. And there are three types of inhibition. First is inhibition of peptidoglycan biosynthesis, and the example is D-cycloserine and phosphomycin. And the main point is the inhibiting the formation of the two basic units, NAG-UDP and NAM-UDP. And D-cycloserine is the terminal 2 amino acid residue of the bearing precursor lipid 2. This is the lipid 2. Consists of D-alanine, and which is produced by the enzyme alanine racemase. And phosphomycin is inactivating the enzyme UDP and NAG-GNO pyruvyl transferase, and it is also known as MURA. And next is inhibitors of dimers comprised of two basic units, or delivery of formed dimers of the growing peptidoglycan chain. And the example is bacitracin, flavomycin. And last, the third inhibitor is cross-linking step that connects chain and between chains, and this is we call beta-lactam antibiotics. And the example is penicillin, cefamycin, and etc. And in this picture, we could see antibiotics. First of all, phosphomycin inhibits the MURA, the enzyme, and bacitracin. This inhibites the PPIase, and PPIase is the enzyme that changes pyrophosphate to phosphate. And penicillin, this we'll see in the next page, more detail. And this slide, we see this slide before. First of all, this we call formycin, and this formycin is also inhibits the NAG and NAM synthesis, and we put UDP on the two basic base units. And same classification, the D-cycloserine inhibits similarly. And doxycycline inhibits dimer conformation, so it inhibits the PPIase. And penicillin inhibits the transpeptidase, so it inhibits the cross-linking step that connects chains. Okay, let's see the beta-lactam antibiotics more detail. And beta-lactam antibiotics has example penicillin and general structure like this. So beta-lactam ring has a 4-membered ring, and we could put 5-member ring or 6-member ring, anything else. And this is the picture how penicillin inhibits the transpeptidase. In the picture, you can see transpeptidase, which helps peptide cross-linking, and when the beta-lactam ring came over to penicillin, they form a covalent bond and prevent the cross-linking. And this is the end of presentation. Thank you for listening.
Okay, as Henry mentioned, this is silver blocking. Anyway, so this is one chain is carbohydrate chain, and they need to cross link like this. This is a small peptide unit. So two mechanisms have this effect. One is making these building blocks. If you block it, then they cannot make this block. So that's one way. Or block this linkage. That's another way. Beta-lactam is which way? Second way. So this beta-lactam is attack these conditions. So this is the one common mechanism for antibiotic. 처음에 중간에 내용 바꾸고 나한테 안 주고 우리는 내가 수업 준비해서 어렵잖아. Okay, so let's check. When we grow the bacteria, when we grow our cells, we grow on the plain bottom and then disperse the cells and then let them grow attached to the bottom like that. If you grow the bacteria, usually we use a soft surface like the agar plate. Agar is the carbohydrate polymer. Anyway, the naming wise, here it says peptidoglycan. So peptido means it is a protein portion or short the protein peptide. Glycan is a carbohydrate motif. And if you change this, you can say glycopeptide or glycoprotein. So how do you define? Usually bigger one comes here. So this adjective form means peptido is a small part, glycan is a bigger part. Glycan can be said saccharide if it's a polymer, polysaccharide, and basic structure is a sugar or carbohydrate. They have different names. These are all carbohydrate structures. By the way, in the middle of I expected the Henry says this pathway too. What is this structure? This is ribosome structure. Okay, ribosome has a bigger unit and smaller unit. Bacteria has a little smaller one, ours has a little bigger, 70S, something like that. But this size is from the centrifuge, the unit. But anyway, this is the higher number, this is bigger. So bigger unit, smaller unit, mRNA passes through here, and then by reading that information, then they synthesize the protein. So if you look at this kind of the cartoon, this is ribosome. Okay. So when we test the bacterial attacking effect, we need to grow the bacteria first. So on the surface of the agar, usually we draw like this, then let them grow. And if it is successfully moving on, then this thin line becomes getting thicker, and then sometimes they form this kind of colonies. If the bacteria forms the colonies, then originally it was an individual of the bacteria, but they start to form big chunks, so-called the colony. In a sense, in other terms, they form biofilm. So once they form this biofilm, now inside and outside they have a different role. Maybe this is the beginning of the symbiosis of multiple cells. If this develops into different function of the cells, maybe that can be starting point of the multicellular organism like us. Anyway, when they form this kind of biofilm, the real shape is like this. They have bacteria inside there, some of them is a dead body, and they have a lot of connecting materials. Proteins, DNAs, kind of materials are coated this bacteria and make it very difficult to detect them, and even antibiotic penetration through this biofilm is also very difficult. So once we have the bacterial infection in our body, they hide somewhere, they form this biofilm, and then they hide there. Maybe they can survive there over 20, 30, 40 years. So the tuberculosis is a typical case. So it's for a long time, they can be hidden inside this biofilm. So to test the antibiotic effect, you grow those bacteria on the surface like this, and then you drop your antibiotic like the one drop, and then this is bacteria growing area, and this is a cleared area. Cleared area is the bacteria was dead. That's why they cannot grow the fast. So that means bigger circle and smaller circle, which one is a more effective antibiotic? Bigger circle, right? So that empty space implies that that drug has some effect to the bacteria. So this is the real example of the picture. So you drop, so this is bacteria growing, and you spot your drug then you can check those of the antibiotics, the activity. Looks like a little primitive, but this is still kind of gold standard method to measure those bacteria growing on real growing. If not, you need to grow them in culture, right? In culture, they are single bacteria, they do not form colony, and they are more vulnerable to the drugs. But once they grow this kind of a colony forming other shape, that they are more resistant. This may be mimic the more realistic situations. Now we heard the story of penicillin discovery, blue fungus or something like that. So if a fungus is growing like that, and most likely if you see, you need to culture either animal cell or bacterial cell. If you see this kind of thing, shoot, this is contaminated by other fungus, flying fungus, the particle. But if you see fungus growing here, and then they see this kind of a clean area, then maybe this fungus may generate some antibiotic material. This is the story of Fleming's discovery of antibiotic, the material. So that material was a penicillin, and that's the basic structure of penicillin is like this. Very nice story has been in the form of a book in English, and also this is translated into Korean. This is good book. How do I know? I translated during my army service. So in this book, there's the full story of how those penicillin was discovered and then developed into drugs. Okay, by looking at those, the bacterial cell wall. Here inside membrane is cell, outside the cell wall. Now those cell wall, peptidoglycan, this is 6-membered structure. This is the carbohydrate structure, glycan part, then connected with a small peptide like this. They have a membrane, and outside of there, there's a lot of the cell wall structure. Okay. Here, whether this carbohydrate structure is very, I said it's rigid and strong, but whether this is fully isolated structure like that, that's the one thing you can imagine. That's why maybe you chose that maybe material transport is limited, but actual structure is not like this, it's like this. As we have seen, those structures were cross-linked, and then those cross-linking has a rigid motif, carbohydrate this way and peptide this way. They make this kind of a structure. It's not fully closed form, but still we can provide this kind of stiff, harder structures. That's why cell wall is not isolated at the wall. It's a kind of just a rigid scaffold there, but that does not block any entry of those any materials. So penetration through the cell wall is a small molecule, and water and nutrients is almost freely. It's almost open structures. Okay, this is the shape of the cell wall, and then by looking at this structure, the carbohydrates and the small peptides linkage like this. Now, already they have been explained. Now, the binding site here is leakage is blocked. How is it blocked? So, this protein, penicillin binding protein, so-called, this is the enzyme which make this linkage here. And then, for that enzyme, penicillin has the shape of a pyrimid here, and then it can bind to the active site here, and then it blocks the function of that enzyme. So penicillin, the example of antibiotic, penicillin is the quite 제가 이거 나중에 바꿔줘. 뭐가 따르면 시간 사용을 꺼야 저기 잡으면 할 때마다 이게 가져오면 되는데. Okay, when I asked you the exciting the example of antibiotic, penicillin is one of those representative first almost miracle drug, and those basic structure is a beta-lactam. Can be said 4-member, 5-member, 6-member. No, it should be 4-member. So this is the amide structure. This is amide, If this amide is a cyclic structure, we call this a lactam. If this is ester and cyclic structure, anybody knows the name of this? Amide, cyclic amide is a lactam. Cyclic ester, this is called as a lactone. Depending on those size, if this is 4-membered ring, alpha, beta, connected to amine. So this is a beta lactam means a 4-membered ring. Then the alpha lactam should be 3-membered ring. This is the alpha lactam. 5-membered ring, alpha, beta, gamma lactam. 6-membered ring, alpha, beta, gamma. You don't have anything finished?
I don't study the Greek alphabet, so I don't.
Okay, 우리가 읽을 수 있다. Alpha, beta, gamma, what's next? Delta, it's a D, right? Delta, epsilon. So that's the direction of the size of the lactam. And why 4-membered ring? 4-membered ring, as you can see here, there's a very high steric hindrance here. Energetically, there's a very energetic molecule. Then why not alpha? Alpha may be too unstable. That's why it cannot be formed. So this is unstable, but it can survive for some time. So this side can be 5-membered ring or 6-membered ring, but this part should be 4-membered ring. Highly energetic molecules. And if it goes into this active site, and active site of a nucleophile attack this position, and this ring is open, and then this enzyme is covalently labeled with this molecule. It's a so-called suicide inhibitor. The molecule goes in and then breaking its structure because of the high energy, and then it's blocked this enzyme atomic. So that's the mechanism of this the penicillin, and general structure of this 4-membered ring of antibiotic. As a whole, we call beta-lactam antibiotics. So beta-lactam, if you hear this term, penicillin-like material. The other side can be 6-membered ring or other structure, it's possible, but key part here, this is the energetic 4-membered ring is the key part of the beta-lactam of the structure. So this is the first discovered penicillin. As you can see here, so this is structure and with the different functional groups around here. Later, depending on those growing of the blue, the fungus that they found there, there's different kind of penicillins there too. It's not single molecule. Original one was benzyl group here. Now later they found that a little exchange structure like this. And this is evolving together. So you may heard first generation penicillin, second generation kind of developed different type. So some of those enzymes they bind to this structure, and then they try to evade the binding or maybe another helpers to break this. So different kind of materials are generated. This is a part of evolution. In that video material that I mentioned that natural product, these guys are generated by fungus. And this molecule is attacking bacteria. Why? That means the fungus and bacteria, they are competing to each other. They wanted to survive and occupy some space, and they don't want the bacteria to come close. And then they secrete these molecules. That means that fungus should not have similar penicillin binding protein. It's a self-killing. So slightly changing those structure and specifically make this molecule only attacking bacteria. That's the key part. And then they both sides they keep changing. So natural product is not direct first metabolite, but this is secondary metabolite, which is not essential for my surviving, but this is attacking somebody else. This is specialized metabolite. This is secondary metabolite. And those materials, now to make this synthetically, it's a little difficult because of this unstable structure. And later, people, the scientists found that if you look at this side, this is amide bond, right? And then by controlling the food to grow the fungus, they could generate this free form. Means by suppressing the supply of this, then bacteria generate this motif. As you can see here, this is intermediate for many other structures. Chemically, now you can modify these positions with many different structures. So that means if you needed to generate this whole by bacteria, the fungus, then this is a natural product. That's a fermentation. But if they can generate, the fungus can generate this intermediate, then utilizing this, you can modify this into many different structures. So this is a semi-biosynthetic the procedure. Then you have much more options to modify those molecules. So this is the beginning of modern antibiotic synthetic drug development. And based on this, it is expanded. Originally, 5-member gene is the penicillin species. 6-member gene structure is the so-called cephalosporin. And then changing this sulfur into carbon. So that is the different type of cover or the pen type of drugs. This is first, second, third generations, expanding those scope and the overcoming those antibiotic resistance. So the mechanism of antibiotics, there are several different the examples as Hyun said. So first, the target you just heard is unique target. Cell wall, we don't have it. Then attacking cell wall is relatively safe. And then attacking those sites, blocking those linkage or the binding, and this is cell wall binding, the cell wall attacking unit. And bacteria is also life forms. Then for them to survive, they need to replicate, DNA replication. They need to express their gene, means they need to make a protein. And if you look at those steps, maybe DNA RNA synthesis attacking, now you should think how their DNA polymerase, RNA polymerase is different from ours. If it's the same, they are already in our side. If you take that drug, then it may kill our body first. So that kind of selectivity is quite challenging. And if you look at the next step, protein synthesis, now this guy comes in, ribosome. Ribosome, our ribosome is bigger. And structure-wise, it's bigger structure and then quite unique structure. So comparing to these enzymes, maybe this RNA ribosome is a relatively easier target to discriminate those shape. So a lot of ribosome attacking molecules has been also used for designing antibiotics. And some specific, the metabolic. So we just heard about the gram-positive, negative kind of things. Is this gram-positive or negative? That gram is how many grams? 2 grams, 3 grams? That gram is the person's name, Dr. Graham. He found that the bacteria, even before they understood the structure of the gram-positive, negative bacteria's difference, by just adding some reagent, colorful reagent, and then they found that some bacteria is stained strongly, the other bacteria is not stained strongly. And then by this, okay, I define, if it's the Hongju made this, then this is a Hongju positive, this is a Hongju negative. Hongju positive is a Hongju negative is So by using the name of that person, and then he just classified this. Later, the structure is discovered, and then realized that gram-negative bacteria, compared to gram-positive, gram-positive they have a cell membrane inside, and they have a strong, very thick peptidoglycan outside, and the staining happened to this peptidoglycan side. So that's why this is stained quite strongly, showing this the purple blue color. Gram-negative bacteria, they have peptidoglycan layer too, because they need some kind of rigid structure outside, but it's much smaller. Also, they have another membrane outside. This is similar to mitochondria. Mitochondria has an inner membrane, and also it has an outer membrane too. Now, the reagent, the chemical reagent, they need to penetrate this membrane. As I showed you, this peptidoglycan structure is with holes. That's cross-linked structure. Physically rigid, but chemically it's almost penetratable to many other materials. That's why the reagent can penetrate inside here very easily. Membrane is tightly the closed form, and crossing this membrane is almost, you need to think about invading new cells. That's why this is quite difficult to penetrate first line here. And then even though they penetrate now the second line, its target is quite small. So this is the structural or the mechanism they elucidated later, almost a half a century later. Gram reagent has been discovered a long time ago, and then why they showed this difference, that understanding took another 50 years. Okay, now bacteria side, I got attacked with a new material, so-called beta-lactam, and that bind to my the important enzyme which make the cross-linking. Now, how to survive? If they survive, there should be some way. That's the problem of antibiotic resistance. For them, it's a fact. They need to develop some way to survive. Then we need to understand how this can be possible. And one of the general question, one of those the resistance is lower their membrane permeability. How? Well, one easy way you can think is third compounds get in, and then you develop new pumps to pump out those materials. So taste bitter. Many materials has a bitter taste. We don't want it to take. Most likely those materials can be toxic material. Then we have some resistance to pump out. Generating pump out. Now decreased uptake. How can you decrease the uptake of the molecule? Special material to make a membrane more rigid or receptor shape changes. Any idea? Jen?
We're talking about uptake into our cells, like what we don't want. Like we don't want the drug to affect our cells, right? Is that what this is about? I think I sort of missed the premise.
So there are several other ways. One is pumping out,
Yes.
and then another way is deactivating this the material. So let's say there was a beta-lactam was the active component. One way is the pump out or the blocking those entry. That's one way. The other way is actively cleave this by other enzymes. Penicillin binding protein is a worker to make this cross-linking, but they generate another enzyme, beta-lactamase. This kind of a new enzyme can be evolved, and then anytime the new the beta-lactam come into the cell, they break it. It's easy to break because this is energetically unstable anyway. So if it's cleave this, and then it doesn't kill itself, just a cleave and hydrolysis of this, then it can be catalyzed for the destruction of those molecules. So physical the removal or destroying the molecule and or the changing those binding target. Let's just say penicillin binding protein, it changes its structure and they do not bind to penicillin anymore. It's kind of evolving, keep generating new type of resistance. So this is some of the examples of the resistance mechanism. So this is beta-lactamase. Beta-lactam structure can be destroyed by hydrolysis and then generating inactive one, and then this can be recycled to attack another beta-lactam structure. Another big issue is the resistance happens in one bacteria. but that beta-lactamase gene can be transferred to neighbors. Even not to the same bacteria, even it can be transferred to different bacteria. Then originally it was a mixture of different bacteria. Only one bacteria has a resistance. It can be spread out, and that's quite dangerous. It's a horizontal gene transfer, and it seems like those genome is inside here, but those small information of the gene can be traveling around. So that's another big issue. Once you have one resistance in one strain, then that the resistance activity can be spread out to others. Okay, so I asked you whether the antibiotic and antiviral. The difference of the antiviral has a clear the name is attacking virus. Antibiotic potentially can mean any materials, but this one is usually, the by convention is usually used as antibacterial. Other materials, they have their own the specific names. Antifungal, that's attacking fungus. Anti-cancer, attacking cancer. Antibiotic is historically used first for the bacteria. So that's why it is antibiotic. If you hear, that's attacking bacteria. Now, big difference between two. Antibiotic, you need to attack those life function, DNA synthesis or protein synthesis or cell wall generation, kind of metabolic attack you need to take. Virus case is a little different the attack you need. They have code, they have the genomic information inside. So you need to think how this virus infect the host cells. So maybe blocking those entry, or maybe destroying their genomic information, or blocking their genomic information copy. So those two have a little different the approaches. So antibiotic drug cannot be used for antiviral infections. It should have a different the drug, different mechanisms. Okay, several questions for LD50. What is LD50? You know? Definition of LD50? Minzela? What is the L? Lethal dose 50. Lethal means killing, right? Lethal dose 50. How do we test the drugs to animals that repeatedly? Other question. LD50 may be different for different species. True. LD50 for mouse, LD50 for the rat. I gave you the example with the nicotine. So some drugs has a more susceptible to some species and than the other. Then how do you use this rat test or the mouse test for human? So that concern is raised over and over again. So that's why the recent days, if we wanted to develop the new drug, we needed to test it, including at least the one rat or the mouse species, but higher animal too, dogs, cat, or ideally maybe monkey. But now to test them, how many animals we should use? That's a big question. So usually you have groups of animals, maybe 10, 100, and different dose. You check how many animals survive by plotting this kind of a curve. And then plot like this. Even though it does not match exactly, approximately take this. Still, whether this can be applied to human, that's not clear. So that's why for human trial, usually we use 1/10, at least 10% of LD50 to be safe. Okay, another question. If it is a solid and liquid, that's easy to measure those amounts. How about it's a gas? If it's gas, how do you measure those amounts? Liquid, you can do maybe micromolar concentration. If it's a concentration, you call C rather than dose. Concentration, molar concentration. Usually when you use the ID, dose, then usually milligram per kilogram. Then this can be applied to solid too. Okay? If it's a gas, how can you do? Still can do like this. Milligram quantity in liter of gas, liter of air. Then how many molecules inside there? It can be used as a concentration or dose. Okay, give an example of a most amazing natural product you can find, the GFP. Yes, I agree. This is one of those exciting material. This topic we will cover at the end of this the lecture later. Okay, so this is the end of today's lecture. Now, from now on, from next week for 3 weeks, we are going to study about anti-cancer drugs in 3 generations of drugs. First week will be chemotherapy, killing the actively growing cells. As one of those convincing example was cell wall attacking, it kills only actively growing cells because already built-in cell wall cannot be effective, right? Only when new cells are dividing and make a new cell wall, that is the point where the beta-lactam can attack and kill the bacteria. Cancer has also a similar the aspect. Fast growing cells will be attacked. That's the chemotherapy. That's next week. The next week, target specific, the signaling molecule attacking is the next week. That's the second generation. The next week will be immunotherapy and cell therapy. So for the 3 weeks of the courses, we're targeting anti-cancer with a different approach and different generations. So if you want to take the chance to present for these topics, let me know by email or call. Okay. Okay. Any question? Yeah.
Test for when
What are they? Is that so important? The max number, max score was a 60. Out of the 60, the average was a 48, and the highest score was 56.
how about individual test?
If you really want to see that, then you can come to my office. After making the appointment and then getting my approval over several range of. If you want it, you can come to my office after this class.